There's a lot to cover. So in the last class, I introduced you to the concept of additive matrices. And those are basically matrices for which a simple tree can be fit, um, given the pairwise distances between matrices. And I gave you the example of this matrix, which basically is uh, if you apply the additive matrix, if you apply the distance-based approach, it would be giving you negative weights. So we basically then go into the idea of actually coming up with an algorithm that can reliably give you the edge weights of the phylogenetic tree. And we do that basically by an algorithm called additive phylogeny. So the idea, the reason why this algorithm failed, the distance-based algorithm failed, was because the shortest distance was actually not the nodes that were having the shortest distance was not actually pairs, were not actually neighbors in this in this graph, and that caused the problem. So to solve that problem, in order to try to estimate the edge lengths based on the linear equations that we had come up with in the last class, we try to come up with the computation of the limb lengths. So limb lengths are basically the lengths of your node to one of the internal nodes, because each node is basically going to be connected to an internal node. Therefore, the limb lengths are essentially the length of one of your nodes to the internal nodes. So the idea here is taking drawing from the equations that we had before. You have these different kinds of distances, right? Values given to you, and in order to calculate the limb length, then you essentially calculate all the possible combinations of orientations of three of your no, basically, um, if you have to if you want to take calculate the limb length of, let us say, I, your final node I from an internal node M, then based on the linear equations given for combinations of two other leaves, right? Two other limbs, two other nodes, J and K. That can be a third node L as well. So you can basically you do calculate limb lengths for all possible combinations, and then you take the minimum one. All right. So the limb length theorem essentially says that the limb length for any node I is equal to the minimum value of minimum value as given by this over all the leaves J and K, right? So J and K, in this case, they were only J and K that were given. There can be many more leaves, right? So all the pairwise leaves you can select, and you can basically calculate the distance, and then you will take the minimum distance. All right. And the limb length problem then becomes we have to compute the length of a limb in a simple tree fitting an additive distance matrix. So input is an additive distance matrix D and an integer J, and the limb length connecting the leaf, basically leaf J to its parent. Remember, the parent is an internal node, is given by limb length J. All right. So how does it work for the example that I shared before? You have these values. Now let us say you calculate, you want to calculate the limb length of chimp, and you consider basically as the common value. Let us say you just take the first one. All right. So now you know that the length of this limb for chimp is 1, right? You know that the length of, let's say, human and chimp was 2, was 3. So basically, you know that so this is 1. So then you can calculate that the human node was 2 away from this internal node. All right. So now how do we actually implement these things in action? So this was the matrix that I was giving you earlier, where you would actually end up with negative weights if you applied the distance phylogeny algorithm, right? So this is the tree that we would finally get, which fits this distance matrix. So what you do is you calculate the limb lengths. You basically start to calculate the limb lengths first. So you pick an arbitrary leaf J, right? So this is J here. It could be I, it could be K, it could be L, right? I recommend each one of you to at least pick another leaf when you do go back home and try to implement this algorithm by yourself and try to do the same calculation. So let us say we pick the leaf J, right? Now we implement the limb length algorithm. We basically try to calculate all possible limb lengths for J, right? So what the answer that we get after implementing this is 2, right? Now that we have calculated the limb length of J, we essentially what we do is we subtract the limb length J, right, from each row and each column, right, of your proposed tree to come up with what's called a bald tree. All right. A bald tree is a tree which is having a bald limb. So a bald limb is basically a limb of edge weight 0. So essentially, because you know that the length of this limb length is 2. Once you subtract the length of this limb 2 from all the other columns, right, including the length of j from m, you get the value of 0, right? So uh, this is how the this is how the matrix would look like. So now once you have this matrix, the next step you do is you now remove the j column, right? You now create what's called a trimmed matrix, because you know that the distance of j to an internal node, some internal node, let's call that internal node M, that is 2, based on the limb length theorem, right? So now if you are having any distances between M and J, you've already removed that part of J that is connected to an internal node 2, right? So you remove that J thing, the J node, because that J node is now the distance of the internal node to j is not contributing to the distance between I and j at all, because the internal node now is going to be lying on I, like in some distance, right? You have basically this internal node now, j is collapsed towards this sort of internal node, right? So you now remove the jth column and you construct the tree called a dtrim. So here things are straightforward. You can basically construct the tree based on the distance phylogeny that was given earlier, giving you basically these kinds of this simple tree, right? And the next thing is, given that you have constructed this trimmed tree, how do you actually attach exactly where on this, where would you actually attach the j? J could be the internal node M. This internal node M can be on this branch, I to K branch. This internal node M can be from I to L branch, right? We don't know, right? In order to solve that, we take advantage of um, some prior information about the limb length J itself. All right, so I'll show you how that works. So briefly, the algorithm was you pick an arbitrary leaf J, you
Sir,
compute the limb length. Yes.
sir, I did not understood how this like a limb length j is equal to 2 coming. Sir, can you please explain again, sir?
Right from this formula, you
Sir,
basically have.
formula I'm understanding, but
So you have to compute now. I cannot show you the computation of simple mathematics here. So I, K, J, right? You have to compute all these things. So, okay. So you have to compute this. So 13, right? So if you want to
yes,
calculate for J, can you apply this formula? What is the formula here?
yes,
IK, IJ minus JK, right? So what is the value of IJ? IJ value is 13. IK value is 22, right? And JK value is, so JK value is 12. right? This is getting subtracted. Ij and Ik value is getting added, right? So
Yes.
13 plus 21, that is 34 minus
34 and
12, that is
yeah,
22, right?
yeah, yes, upon 2. So that will be 11.
That will be 11. Now you will
Yeah.
also do it for L, right? You will do for all the different pairwises and you will get the value. So let us say you do it with K and L. So it will be 21 plus 12 minus 22, right?
Yes.
So like that, you will have all the different combinations and it will be 2.
Okay, sir. Okay.
Yeah, do it by yourself and you will be able to find it.
Okay, sir.
So this is the algorithm. So now you remove this jth row, this arbitrary row that you took, and you create this trimmed matrix. All right. You have the j bald here. You construct the tree of this trimmed from the trim matrix. And now you have to identify exactly where in this tree you should attach the leaf j. And once you have attached the leaf j, you already know the limb length of j. So you can reconstruct the tree. Now the important point is how do you identify where to attach j? Now there are the limb length algorithm, limb length theorem again comes into our rescue. So we already know from the limb length theorem that if you want to calculate the value of the limb j bald, right? For this basically for this matrix in the so for this matrix d bald, if you want to calculate the limb length of j, the limb length of j will be given by this particular minimum value of this, right? Over all the possible combinations of j and k, right? So when you find the minimum value of over all the possibles of j and k, so here it could be jk, it could be j and l, it could be k and l. All right. When you do this for all the possible values, then the minimum value that you actually get is 0, right? Because this is if you yeah, if you do it with L and I or K or L, the minimum value is going to be different, right? So the minimum value that you will actually be getting is when you consider I and K. So when you consider I and K, the minimum value is 0, which essentially means that uh, basically the length of ij plus length of jk is equal to the length of I and is equal to the length of I and k, right? Which essentially means that the point, right, at which you have to do the attachment, it lies between I and K. Is this clear to everybody? Is this explanation clear to everybody?
So it's clear.
So the attachment point, basically J is found on the path between the leaves I and K at a distance of I comma J, right? I comma J is already given to you. Sorry, not from the Bald matrix. This value is given to you, this value is given to you, and this you basically extrapolate this value from here. All right. So this is the basic idea of additive phylogeny. And this given an additive matrix, if a simple tree, basically if the matrix is additive, you can implement additive phylogeny and can calculate how the tree would look like. So now if you look at the protein distance matrix, so spike protein is the protein that is found in on the coronavirus. Um, or different coronaviruses that infects this respective organism. This is how the matrix looks like between the based on the alignment scores between the pairs. So if you would look at this matrix, can you tell which organism was responsible to give us SARS-CoV, the first version, SARS-CoV-1? I mean, this is, you don't have to read the slide here to answer that question, right? What would you do for that? Which
Yes,
spike
civet.
proteins, right? Civet and human is the closest one. So you would suggest that um, civet and human would be giving you that because the coronavirus is very similar. So it must have jumped from civet into humans. But this would only be true if the matrix is additive. But the matrix actually, this one is not additive, and that's a problem. So we can make the matrix additive, and then so you can basically construct a tree. But yeah, once the matrix is additive, then also it's possible that they may lie on different arms, right? But because the distance is low, most likely in most cases, that when the distance is very close to each other, this is where you would say that some bacteria or some virus jumped from one organism to the other. All right. So 16 is the lowest one. And when you actually reconstruct the phylogeny from this additive version of the previous matrix that I gave you, this is how the phylogeny looks like. This is the root area, and this is all the present species of coronaviruses that are infecting the respective organisms. And you see that palm civet and human are close by in the tree. So this is how the palm civet looks like. It's actually harvested for its meat in China, as I said in the last class, and therefore that's what's suspected, that that's how the virus jumped over. And for the current outbreak, it is suspected that either it was bat or it was another organism called pangolin, which was the coronavirus had sort of jumped from these organisms. So the question is, in the previous case, in the previous algorithm, in order to actually have a tree, you need to have a matrix which is additive. But what happens, or how do we actually work with matrices that are non-additive? So in order to do that, we come to another set of algorithms, which is called ultrametric evolution trees, which ensure that given a matrix, a tree is fit into the matrix. So in order to motivate the algorithm, I have to talk about a few ways in which researchers modeled speciation. So researchers basically assume that all the present day species are derived from some ancestral species that split at some point of time into two. All right. And whenever an ancestral species, as represented by one of these internal nodes in the tree, these species, whenever they split, the same time has passed from the splitting event to now, and the rate of evolution along that splitting event from the species, the ancestral species to species now, the rate of evolution is exactly the same. So you can imagine that the distance between the species currently and the ancestral species is the same between the organisms that are observed now. So what do I mean by that? So this is, for example, a speciation tree. You can model it like this, right? But this particular tree is a unrooted tree. It's an unrooted binary tree. So every node has either degree 1 or degree 3. So the leaves are having degree 1 and the internal nodes have degree 3, right? But in this case, we are not basically able to tell which is the ancestral species, or exactly how these things are sort of related with respect to time, with respect to evolution. So these kinds of trees have given way to what's called rooted trees. And in a rooted tree, you essentially have a root, right? So in these other trees, you did not have any node which was the ancestral node. So somewhere on these internal edges, you put another node. You take this node and you pull it up, and then everything else falls down. This node, which is of degree 2, is called the root, right? And then this tree, this unrooted binary tree, becomes a rooted binary tree, where the assumption is that this root represents the furthest most ancestor of all the species, right? And all the internal nodes now represent speciation events whereby the species, ancestral species, was split into two kinds of species. And all the species which are currently observed are species which are having the same amount of time, that have taken the same amount of evolutionary time with respect to the previous ancestral species. So what do I mean by that? So for example, if we look at this internal node, so this internal node represents an ancestral species which split into chimpanzees and bonobos at exactly the same time, right? So the length of these limbs is exactly the same. Similarly, if we talk about this represents an ancestral species of monkeys, which later on which split into a lineage which became humans, and another lineage which finally became chimpanzees and bonobos, right? The idea is that the length of the root, right, this root and all the internal nodes is exactly the same to all the leaves. The assumption is, and this basically allows you to assign a molecular clock, right? So the topmost node is the earliest node, and let us say its value was 33 in terms of the number of mutations you would require to reach this root node, and then you further down, you go down, you have more and more mutations. So you basically see that from going from this root node to the squirrel monkey, 33 mutations were required. From going from this root node to this internal node, 10 mutations were required, right? But again, going from this original uh, original species all the way to squirrel monkey, baboon, orangutan, gorilla, chimpanzee, bonobo, and humans, all of them are equidistant. All of them should require 33 mutations to happen. And the number of mutations, they can, researchers basically can look at other characteristics of the organisms or mutation rates in general, and they can come up with they can convert these mutations into molecular clocks, right? So when we say molecular clocks, all the species which are seen now, they have age 0 because they are currently present. And these ancestral species, which seem to be mutated by a certain, certain degree, they represent evolutionary times which can be converted into, say, millions of years. So this 33 mutations can be corresponding to about 3 or 4 million years of evolution. All right. So now how do we actually construct these ultrametric trees? The edge weights of these trees, as I was telling you, represent the age between the nodes. All right. So this is the age between the nodes. And this in ultrametric trees, the distance from the root to any leaf is the same. That's the idea. And the assumption is that the evolution rate along each branch is the same, right? This is one of the biggest assumptions in such ultrametric trees. So that was a very complicated explanation of what these things are, but they are much easier to make. And I'll show you how one actually makes these ultrametric trees. So the method that you use is to make ultrametric trees is called UPGMA, which is essentially, I forgot the full form. Just give me a second. So it's unweighted pair group method with arithmetic mean. All right. So you essentially use arithmetic mean to figure out the lengths between the branches uh, from of the leaves from internal nodes. You take arithmetic mean because the assumption is that the evolutionary rate across the branches is exactly the same. I'm emphasizing on this idea again and again because this will be important for the next set of algorithm that we'll be doing. So here, let us say you are given a non-additive matrix as shown here. So this is the one matrix that I gave you earlier. This is a non-additive matrix. And now we actually use one of the sort of a greedy approach, something that actually Bhavya pointed out, that let's start with nodes that are having least distance between each other. All right. So which are the nodes which have the least distance between each other? So they must be close to each other, right?
And
Right, K and L, right? So what we do is we assume that K and L must be connected. Now, if they are connected, that essentially means that the distance from them to an internal node, right, is going to be equal. So what you do, you put an internal node which connects K and L, right? And the distance between K and L now gets distributed across the from basically from this internal node to these leaf nodes. So this distance 2 is distributed 1, 1, right? And this internal node now has a distance of 1 from these leaves. Now, once you do that, you essentially converge this I as this K and L together, right? So now distance of all the nodes, all the other nodes, so I, J from K and L can be calculated by taking the arithmetic mean of these columns. How does it look like? You essentially take the arithmetic mean of these two columns, right? So it's 3.5. You take the arithmetic mean of these two columns, right? So basically K and L are combined now. You take the arithmetic mean of this and this, so it becomes 3.5. I and J remains the same, right? So this is how you update the columns now. Now you iterate over this. Now the next shortest distance is I and J, right? So you put internal node I and J. Do you distribute this distance 3 arithmetic? It's because it's the arithmetic mean, so it's distributed to 1.5 on both sides. And this node, internal node is 1.5 mutations away, let us say, take some metric away. Now you again collapse. Now these two things are collapsed, right? And now you calculate the distance. So in order to collapse them, right? Now you actually again calculate the distances by taking the arithmetic mean of these two things, right? And these two. And this is how you will, this is what you will be getting. Okay. Now again, you know that I and J and K and L are themselves 4 apart. So now you would have put an internal node, which is basically distance 2 apart, right? But these internal nodes, you are talking about I and J and K and L all the way from the root to this area, right? So from this node, the top node is 2 away from the bottom node, right? So you distribute the distance 2 all the way down on both ends, right? So this node is 2 away. Now, given that this node is 2 away, this internal branch becomes 0.5, this internal branch becomes 1. Right? And you basically iterate over it. So the idea is you form the cluster of present day species, right? Each containing a single leaf. You found the 2 closest clusters, right? You average the distance of these clusters. You obtain the internal node and you distribute the distance of this internal node. Then you merge cluster C1 and C2 into a single cluster. The new node is going to have different edge weights, which can be calculated by taking the averages of the existing edge weights. You update the matrix by computing the average distance between each pair of clusters. And then you go back to the second step, right? Until all basically you are left with just a single node where everything has been combined. Is this clear? This method?
Yes, sir.
Yes, sir.
You combine this, you get this, right? For going from this matrix to this matrix, this is clear. So I and J is unaffected, right? But K and L, you take the average of this. So this is becomes 3.5, 4.5. And these two things, they are just merged, right? So you don't have to worry about them. The distance between K and L is the same. Similarly, you merge them, right? Distance between I and J doesn't matter, but the distance between I and K and J basically is this, right? So distance of K and L is 3.5. Distance of K and L from the merge is 4.5. So you merge them, right? So this becomes 4. Similarly, 3.5, 4.5, you merge them and this becomes 4. All right. So
Yes, sir.
now if you actually use this to implement the on the original matrix that was given to you. So this is how it looks like. So instead of now having edge weights, right, sort of the limb lengths, you actually have the ages of the internal nodes. And this tells you that the mutations, the ancestral strain that could infect both, that was diverged, that could infect the palm civet, had 8 mutations, and that could basically infect the humans, had 8 mutations. So just like that, this is how this is how this particular part of the virus was evolving. Right. So now again, whenever I say code challenge, think about writing a pseudocode to implement the specifics of this particular thing. So when I say pseudocode, it's not writing these things, but it's actually given a matrix. Actually say how do you extract the column, how do you extract the row, and give a code for doing that. All right. But if you look at it, does this given tree actually fit the matrix? So is the distance between I and L 3?
No, sir, it is 4.
It is 4, right? So we know that the matrix is not additive, right? So a simple tree is not going to fit it, right? So the basic idea here is that this idea of the tree fitting the matrix is only sort of valid for additive matrices. It doesn't hold for non-additive matrices. So the whole idea of UPGMA is to make sure that a tree fits to a matrix. It doesn't have to actually sort of a tree basically can be made from a matrix, but it doesn't necessarily represent the real distances that you would find between the sequences. So now, what is the problem here? Right? So let us summarize what we have learned so far. So in additive phylogeny, it's good because it produces a tree fitting an additive matrix, but it is bad because it completely fails for non-additive matrices. You get negative edge weights and you cannot move ahead. UPGMA, it's good because it will produce a tree for any matrix, right? So it doesn't matter whether it's additive or not, but it is bad because the tree does not necessarily fit an additive matrix, or it does not necessarily represent the evolutionary distances accurately between the species involved. So we basically need to come up with an algorithm that produces a tree fitting an additive matrix, and that same algorithm also uh, fits or sort of produces a tree that is very close to what a non-additive matrix distances represent. All right. So this algorithm is called the neighbor joining algorithm, and it actually works on a transformation of the distance matrix that is like magic. So this is the transformation. So given an n cross n distance matrix D, its neighbor joining matrix D star can be given by the following expression. So n is the number of nodes that you're having or number of leaves that you're having, right, in the tree. Dij is the distances that you've already been given. And now we basically introduce the idea of total distance di and total distance dj, right? So the total distances is the sum of distances from I to all the other leaves, right? If you were talking about total distance I. So this simple transformation of the matrix actually converts the non-additive matrix to a more additive version. And it helps you to actually solve solve the phylogeny using the same ideas as additive phylogeny. And I'll show you how. So let us say you had this matrix, right? Earlier, if you remember this matrix, if you apply the distance additive phylogeny or distance-based methods, this gives you a negative edge weight, right? Because this matrix is non-additive. Now what you do is, in order to convert this into D star, right, this transformation, you first have to calculate these total distance DIs. So the total distance di is going to be the sum of distances of this I node with all the other nodes. It's 13 plus 21 plus 22, right? So you basically calculate the distances. So this is 56 for the case of I, for the case of J, it's 38, for K it's 46, and for L it's 48. All right. Now the D star IJ is basically shown here. All right. Is this clear? Yes, no?
Yes, sir.
Yes, sir.
Right. So now the neighborhood joining theorem basically is that if D is additive, right, then the smallest element D star corresponds to the neighboring leaves in the tree D. Right? This is a theorem. It's quite a big proof for this. It's out of the scope of the course right now, but if you're interested, I can share the links where you can read more about it. All right. So for a matrix D, the smallest element of the D star corresponds to neighboring leaves, and is actually true not for just additive matrices, but it's also true for non-additive matrices. So then in this case, in the case of D star, the closest neighbors would be I and J and L and K, right? Earlier, the closest neighbor was actually J and K, right? You see the difference? So in the case of D, the closest neighbor was J and K, but after you have done the transformation, it suggests that the closest neighbors are actually I and J and K and L. All right. So what are the steps for implementing this algorithm? First, you construct the neighbor joining matrix D star from D, right? Then you figure out what are the minimum elements of D star, right? Which were the minimum DIJs, D star IJs from D star, right? So in this case, it's IJ and KL, right? So let's say we pick one of these, we pick IJ here. All right. After that, you compute the distance of the limbs between, so the distance between ij, right? And this distance between ij is written as delta ij, and this is given by total distance I minus total distance j divided by n minus 2. All right, so this value is 9. Now, in order to figure out the limb lengths now, all you have to do, you have to set the limb length of I as half of dij plus this distance, right? And limb length of j as half of dij minus this distance. Okay, so you get these values. Now you do the same thing as we did with the first additive algorithm. You just remove I and j. In this case, you replace it by an internal node. Let us say that internal node is m, right? So now we know that the limb length of I and m, so basically the length IM distance is 11. The distance MJ is 2, right? So we remove these things and we replace it by M. And we can apply the same ideas as before to calculate the distance of M from all the other remaining nodes, L and K, using the limb length theorem, right? And you again have the total distances. You can keep on repeating this idea, right? And you will be able to now. So basically, given there's these things, you can calculate these distances again, and you will be able to now actually make the matrix. You do the same thing. You apply a neighborhood joining on the D prime. So this D prime matrix. So you to obtain the tree for this D prime matrix. You will apply the same ideas as before, and you are going to get the tree. Right? You already know that the limb lengths are here like this from the internal node M. You know that internal node M has these certain distances, and they are distributing as 10 and 11 like this, and you're going to obtain the tree. Right? So this is the algorithm for it. You construct a neighborhood joining matrix. You find the minimum element. You compute these delta ij's. Using these delta ij's, you set the limb lengths of limb length of I and limb length of j by the given formula. You construct just like you constructed a bald matrix. So you construct, so not a bald matrix, the first matrix that we were talking about in the distance method. You construct a matrix D prime by removing the ith and jth row and by adding the mth row and mth column such that for any value of k, the value distance of dkm is equal to uh, basically this distance given from this formula, right? And you apply this neighborhood joining again to the D prime graph, and you do it until you are able to attach all the limbs into the tree. All right. This is slightly complicated. Although I am saying code challenge here, I would not ask you a question to implement this for sure. So now, a homework problem that I expect all of you to be doing is to implement the same, is to implement the neighborhood joining algorithm to the same matrix as was given here, right? So here the matrix that I used was this one, right? But I want you to use this neighborhood joining algorithm on this matrix and tell me how the tree looks like. All right. So this is how these if you apply neighborhood joining to the coronavirus, the non-additive matrix, this is how it will look like. Now you would see a peculiar difference here. Can somebody point me out the peculiar difference between the graph that we are getting here, the tree that we are getting here, and the tree that we got with UPGMA? Anybody?
There the distances were the same.
Sir, the lengths are bigger. Like in previous tree, the distance between palm cuvette and human was smaller. Like it was, means they are closer to the this right hand side. Means the
Also,
node values are
the
bigger.
equidistant in that one.
Is the distance between the internal node and the leaves the same?
In the other one, it was the same, as in both were equal, whereas here it is different for both the leaves and the internal node.
Right? So each leaf actually is having a different distance from the root or the internal nodes.
Sir,
So
I guess
which
this is based on like how much mutations has happened actually, like uh,
yes,
from yes, sir.
yes.
Like
So what does this mean?
from pig and cow, there may be one ancestor, and from that ancestor, like 146 mutations is required for cow, and 149 for pig. But on the
Exactly.
when we were seeing that first one, that was only based on time scale.
Exactly. So the idea, the difference between the two algorithms is in the case of UPGMA, the assumption that the evolution, the rate at which or the time at which the species came about, right, is the assumption is that the evolution is happening at the same rate on each of the branches, right? That is the assumption in the UPGMA, but that is not the case in neighborhood joining because you see that the branch lengths are different. So typically in neighborhood joining, you will see the cow is actually going to be slightly on the left side here than the pig, right? Because the branch lengths are different. The mouse is going to be slightly, so these things are, this is how these trees will slightly differ. So now if we implement this neighborhood joining on the coronaviruses that were isolated from different patients at different time points, so you could actually see that the first coronavirus, the closest coronavirus that was from Palm civet was isolated from villagers in the Guangzhou district of China, which was then passed on to the neighboring district. Then from Guangzhou, it was passed on to that doctor who had looked at the virus, attended to the virus, and then had gone to that hotel. And then from that hotel, it was basically spread out. So you have Hong Kong, it went to Hong Kong, it went to Hanoi, it went to Zhongshan, some other village. So most likely this was the last place where he had come across the patient, right? So it went from Palm civet to Guangzhou. it went to Zhongshan. Then from Zhongshan, it was probably the doctor looked at the patient from Zhongshan. From there, doctor contracted it in Hong Kong, and from Hong Kong it spread to Toronto and to Hanoi. All right. So this is how these kinds of trees help you interpret how these kinds of outbreaks happen and how you can actually trace the origin. And again, you can see that the lengths of these different limbs are not always the same. Is this clear to everybody?
Yes, sir.
Yes, sir.
So although they are great, these distance-based matrices have one big problem. The distance-based algorithms for evolutionary trees, basically, they are not telling you exactly which nucleic acids or which amino acids were mutated, right? So exactly what was the status of these internal nodes of these ancestral species is not known by this. You only know that they were differing by a certain number of nucleotides or amino acids, right? But you do not know where those mutations were happening. But we know that evolution doesn't work like that. Evolution works on sequences. So if we say that an ancestral species was existing, right, that essentially means that it must also be having some kind of nucleic acid or some kind of amino acid sequence. All right. So whenever we convert an alignment into a distance matrix, we lose the information about the underlying sequences. And this is one of the biggest weaknesses of distance-based matrices. So in order to solve a distance-based methods, so in order to solve this problem, there you come up with what's called character-based tree reconstruction. And this is one of the oldest ways in which evolutionary biologists have been looking at or basically telling constructing phylogenies is actually by looking at characters. So the characters could be for us, these characters could be, let's say, easy physically recognizable characteristics. Let's say whether an insect has wings and whether an insect has how many legs that insect has, right? So these are characters, wings and legs. So you can, for example, I'm showing you 3 insects here. One insect has wings, 2 insects are wingless, 2 insects have same number of legs, and another insect has many more legs, right? So now based on these physical properties, these anatomical properties, you can also make phylogenetic trees. So if you want to make a phylogenetic tree based on characters, the idea is to reconstruct the phylogeny, right? You are given n cross m characters. N is the number of species you have, and m is the number of characters you have. The output would be a tree in which species with similar characters are occurring close to each other. Now, if you ever think that biological algorithms and problems are not interesting, you would be surprised to know that similar character-based phylogenies are actually used are the basis of a lot of these YouTube and Netflix recommendation algorithms, because they are also trying to see who is watching what kind of content and then suggesting that maybe people who are watching two similar kinds of content might also like to watch content that they have not, that two people haven't seen, right? So for example, in class, let's say Dev and Bhavya are watching similar kinds of movies, right? And one movie Dev hasn't watched, that will be recommended to Bhavya, and one movie Bhavya hasn't watched, that will be recommended to Dev, right? So these are also, these kinds of character-based phylogenies are really useful not only in biology but beyond that. So if we try to do this kinds of character-based phylogenies for, let's say, coronaviruses, right, which are microscopic creatures, do you think it really makes sense?
Sir,
How
I
many
don't
characters?
think, sir, it will make sense. Like in class 10th, like something was there like analogous and homologous. So there was a difference between that. So uh, if this is homologous, then definitely we can, but not always, right?
Sure. But my question is actually, can you look at, would it be reasonable for you to actually look at a lot of the physical characteristics of coronavirus? What all physical characteristics you can get from this picture?
We can look at the spike protein and the shape of this virus, and
Right?
I don't feel
But
more.
it's
Sir, I
not a,
think, sir, horizontal gene transfer might also cause issues.
Sir, horizontal gene transfer happens even in virus.
horizontal gene transfer is completely different. I'm just talking about physical characteristics right now.
No, sir, no, you can't really compare physical characteristics of microorganisms.
I mean, people have been doing that, but it's a tough job, right? Because you need to have a
Yes.
microscope every time, and you need to actually get physical. So let us say shape and the number of spike proteins on the coronaviruses are, let's say, the physical characteristics that you want to do, right? That's not a lot to distinguish between different coronaviruses. So therefore, people actually would want to construct phylogenies not from physical characters, right? Like wings or wingless, but they actually want to do this from nucleotide or amino acid sequences. When you actually do this from different characters, there is actually one problem also that has been seen. So it's perfectly reasonable to assume that if you would make a phylogenetic tree, the winged insects and wingless insects are going to be separated, right? Is this clear? Would everybody expect the same?
Yes, sir.
Right? So this is
Yes.
actually called Dollo's principle of irreversibility, which basically says that evolution is smart. Once it has invented an organ such as insect wings, it's not going to reinvent it again and again, right? So basically, the assumption is that whenever you make a tree of insects, the winged insects and wingless insects are always going to be separate. You are never going to as find winged and wingless insects coming together in the phylogenetic tree. But what people actually saw when they made these phylogenetic trees was only for a very small group of insects, that even in the small group of insects, you actually saw cases where winged and wingless insects were getting categorized together. So for example, this is the location where, you see, where the ancestor was wingless, and now you have winged progeny. Here is another example. Here is slightly older example when this happened. This is more younger example. This is also same time as this one. This is even further ago, right? The super ancestor grandparents were wingless, then children were winged, then their children were winged as well, but one of their children, grandchildren, became wingless. Right? So in evolution, this not necessarily is always the case when you are talking about physical characteristics. I think this is what you were talking about, Ratnesh, about homologous and non-homologous characters.
Yes, sir. Yes, something like this. This
So
like
yeah, so therefore it's not a good idea to actually look at these trees from construct these trees from um, physical characteristics. Therefore, people have suggested to actually make these phylogenetic trees from actual nucleotide sequences, because this is what the substrate of evolution is, right? Evolution is happening on nucleotide sequences. So now what you have, instead of having n species and m physical characteristics, you have n species and m characters of nucleotides or amino acids. All right. So what do we mean when we talk about making character-based trees? Can be summarized as a parsimony problem. All right. So if you are given sequences and alignment like this with n species and m characters, you essentially want to organize these species or these sequences in a rooted tree. All right. Such that based on this rooted tree, the sequences can be used to create consensus or basically uh, ancestral characters as well. So for example, if you have this kind of an organization of species, this kind of a tree, this is what we want. We want to have the ancestral states of the internal nodes. All right. So when you have the ancestral state of internal nodes, you can actually now take a look at the Hamming distance of each of these internal nodes with the leaves, right? And you can come up with the weights of these edges. So earlier we were inferring the weights of the edges just based on distances, not based on the sequence itself, right? But here now we essentially first infer the sequence and then we calculate the distances. All right. This is a difference because some sequences or some kinds of mutations might be occurring together, which might be easier to occur, right? And some mutations might be occurring randomly all over the place, which might be more tougher, right? So they might be scored less favorably. So in this case, the score or the parsimony score for this kind of a tree is given as the Hamming sum of Hamming distances along all the edges. All right. So when you say you want to have the a good tree computationally, so you can summarize say it as a small parsimony problem. So in this problem, the idea is to find the most parsimonious labeling of internal nodes of a rooted tree. Input is a rooted binary tree where each leaf is labeled by a string of length m, right? This tree you can get from any of the previous methods that we had. Now, after this, you want to provide the most parsimonious labeling. So you want to give the labeling of all the internal nodes, right, of string length m, such that it minimizes the tree's parsimony score. So that means there are least number of mutations that are going from the parent node to the root nodes, right? So sorry, from the root node to the internal nodes to the final leaves. That is those internal uh, sequences is what you have to find out. All right. Can we simplify this problem statement? So before I leave the class today, can you simplify this problem statement that you can, if you can solve that problem statement, then you can also solve this problem statement. So you are given a rooted binary tree with each leaf is labeled with a string of length m. The output is labeling of all the nodes of the tree by the strings of length m that minimizes that tree's parsimony score.
Shouldn't it be like consensus string?
It does not have to be the consensus string always, because in this case, let us say you have a mutation, right? This is a mutation. Let us say this is CCT right here. This is ACT. So the consensus string is going to be what? You're only having 2 for each node. Should it be a C or should it be an A? Right? Because this is an actual organism. You don't know. It has to be either a C or an A. Do you understand, Bhavya?
Yes, sir.
So it actually has to be one of the letters. It cannot be the consensus sequence. Consensus can only be when you have uh, multiple sequences, right, that are aligned. A simple version of this problem statement would be if we can solve this problem for a single symbol, right? What do I mean by that? Let us take only a single symbol here, the fourth column, right? This is an A, this is a T, this is a T, goes to T, this goes to a T. This is an A, this goes an A, and in both the seal and whale you have a A. All right. And there's a dynamic programming algorithm which we can use to solve this. This is what I'm going to discuss in the next class. All right. I'll stop. Any questions? I'll take.